A very fresh and hearty afternoon to everyone present here. I hope all of you are doing great. I, Vanchika Garwal, feel highly honored in extending my warm welcome to everyone attending this very interactive and knowledgeable webinar on the International Mangroves Day. I must mention that this is a great initiative taken by the Eco Club of Tecnia Institute of Advanced Studies to aware the students about such important days. So let's not take much of your time and please invite the Honorable Director, Sir, of Tecnia Institute of Advanced Studies, Dr. Ajay Kumar, for his ingenuous words. Please, sir.
Uh, First of all, I would like to thanks the Eco Club that you have taken yet another initiative on the issue of the Azadi Ka Amrit Mahotsav, the mandate which has been given by the Eco Club of Government of NCT of Delhi, further to celebrate the 75th year of our independence of India. The mandate given to us of UN International Mangrove Day. So the moment we have been working on this mandate of what is International Mangrove Day, most of the students have been saying that what exactly is the mangrove, and reason being the faculty concerned over here, Dr. Nivedita and others, those who are from the biology side, they are the botany side, they will be able to identify what is the relevance of mangrove. So to make the student aware what exactly is the mangrove, International Day for the Conservation of Mangrove Ecosystem. It's not just a lifeline for the coastal communities. UNESCO is also celebrating the International Day for the Conservation of the Mangrove Ecosystem every year with the aim of raising awareness about the mangrove ecosystem. According to the United Nations, UNESCO, mangroves are disappearing at the rate of at least 3 to 5 times faster than the overall losses of the global forest, and which is the cover of the face for the infrastructure development, urbanization, agricultural land conversion. And when the cyclone Amphan hit the Bengal Sundarbans region in May 2020, it left a trail of destruction reminiscent of the 1999 super cyclone that killed thousands in Odisha. Experts rued that Amphan devastation would have been limited if there has not been a rampant deforestation. So human activity, deforestation, including tourism and fishing, and changes in the land were reduced the health and the extent of the Sundarban mangrove cover. So Sundarban is a home for the world's largest mangrove cover. One of the India is having the world's largest mangrove cover, and a prolific ecosystem between the land and the sea and the ocean. The coastal communities all over the world depend on these ecosystems for their well-being and protection and for the food security. So they are the breeding grounds for the, you can say, several, you can say, marine organisms. And biggest contribution towards these coastal communities is the act of the acts as a natural barrier against tsunami and storms, surges, and the rising sea, sometimes erosion also. So the Kyoto Protocol has rightly put on that we have to create among our younger generation the sense of sensitization towards the environmental factor. So climate change is one of the most important factor. We are talking about various global warming and the change in scenario. Of late, we've been seeing that the mountains are crippling and our national highways were lost in Himachal Pradesh. The Chamboli, lot many things is uprooted because of these human activities. So mangroves is one of the important areas which can be able to make you near to the natural system, natural ecosystem. Without taking much of your time, I feel that we have got our experts with us who will be able to tell us what are the current estimations of the mangrove coverage in India and how the things are shrinking for the last 40 years, and what we are doing and what it is expected out of us to safeguard these mangroves, including the Godavari Krishnan mangroves, Batiknath mangroves wetland, Barthang Island, Andaman Nicobar wetland has been also there. And lot many mangrove systems, ecosystems are over there in India, including Tamil Nadu, including Goa and some areas, coastal belt areas. So government of India has made its efforts that total mangrove cover should increase, and they are making this it sensitized that each and every student of BA, BBA, and BJMC will be able to cover these topics in time to come because it leads with the ecosystem. So, climate change report and the Forest and the Environment Ministry, Union Ministry of Environment and Forest and Climate Change reported certain things in line of that mandate of Eco Club, which has been exerted by the by our government. We further we have to preserve our global geoparks, world heritage sites of various biosphere reserves for the future generation. That's very important. And we have to make and promote these sustainable management and conservation of these mangroves. With this, I feel that they will be able to highlight that what are the importance of mangroves, how the forest cannot be overestimated or underestimated. If these are the natural barriers and natural disasters, maintaining the natural disasters for the centuries for the conservation of the same thing. We feel that the experts over here will be able to give you right insight about the mindset of the what exactly is to be done in this area. Thank you very much for this giving me opportunity and making me part of these change factors, and each and every student will become a change agent in time to come. At least they will be able to give awareness to other people that what is the role of mangrove over here, and think on it and discuss on it on various portal and on various blogs on social sites, and make the general awareness among the NSS and the Eco Club. Good luck, and you will be able to achieve your objectives with the right thing. Thank you very much.
Thank you so much, sir. Your words always give us new insights and overlooks about such unsung topics. So now it's time to please invite Dr. Nivedita ma'am, the, the Training and Placement Head of Tecnia Institute of Advanced Studies. Ma'am, please address the gathering.
Thank you so much, Vanshika, for giving me this opportunity to share my thought. Uh, uh, I will just share some of the slides which I have just prepared for you students to understand what is my understanding about mangrove and how I perceive the things for you. So I'm just presenting my presentation here. So I have just tried to put my thought for this particular day celebration, that is International Mangrove Day celebration, what we are observing it today. And I tried to focus that to me what is mangrove and how we are how we are impacting this mangrove and why it is very important for us. So this particular picture, it is for Royal Bengal tiger that it has been taken from Sundarban, and as Dr. Sir has already said that Sundarban is the largest mangrove forest of the world, and it is also a UNESCO heritage site. So if you talk about mangroves, what at all are mangroves? They are salt tolerant vegetation that actually grow in the regions where tide comes and where estuaries are there. So they are also referred as tidal forest or sometimes wetland rainforest ecosystem. Right? This particular picture, it is from Florida's mangrove forest. It is written here. If you talk mangrove ecosystem, it is the interface between the terrestrial forest and aquatic marine ecosystem. It includes diversified habitats like mangrove dominant forests, little laden forest, floors, mud flats, coral reefs, and contiguous water. Right? These are trees. Mangroves are actually trees and shrub species that grow at the interface between land and sea. And what the uniqueness of these trees is that they have a specialized root, and these roots are known to absorb. They come up above the water and take oxygen from the air, because the roots, they are always immersed in mud or soil, and because of that the water, the excess of water in the soil make it the oxygen deficient system, and that is why the roots come up above the water and they take oxygen from the air. Now if we talk about they create a very unique environment, and very typical environment which is very much particular to this particular system only. And if you talk about the number of fish, number of crustaceans, for example, I mean turtles and crabs, they used to grow in this particular ecosystem only. Have you heard about this particular species, red-lay turtles, which is now endangered? This is one particular turtle which is very much common in various mangrove forests. Now, have you heard about any fish which can climb the tree? This is tree climbing fish. This is again found in mangroves. This is another example which is in mangrove forest only. Proboscis monkey, kingfisher, again a famous name. So if you talk about that, it very unique type of organisms and plants are found in mangrove forest. If you talk about India, how the mangrove forests are distributed, you will find that the highest area under mangrove forest is in West Bengal, followed by Gujarat, Andhra Pradesh, this is Andaman and Nicobar Island, and Andhra Pradesh. So West Bengal has the highest kilometers area under the coverage of mangrove forest. Now, if you talk about this West Bengal, we know that in West Bengal we have this Sundarban, and this is UNESCO World Heritage Center as well, and is the largest mangrove forest. If you talk about where it is in the Gangetic Delta, and it is very important, universally important for globally endangered species, including Royal Bengal tiger, Ganges Irrawaddy dolphins, estuarine crocodiles, and the critically endangered endemic river, that is Batagur Baska. It is the only mangrove habitat in the world for Panthera tigris, that is the Royal Bengal tiger, right? So why mangrove matters? I'm just quoted here saying from one of the fishermen from the Thailand, if there are no mangrove forests, then the sea will have no meaning. It is like having a tree with no roots, for the mangroves are the roots of the sea. So this tells the importance of mangrove, and especially for those people who are actually living near the coastal area where the livelihood depend on the sea and the marine ecosystem. So mangroves are like kindergarten, sea grasses are the secondary schools, and coral reefs are the high school and colleges for fishes. And once the fishes graduate from university, they return to kindergarten to spawn. This is again saying by Khun Fishet. He is co-founder of Thailand's Yat Fawn Mangrove Preservation Project. And this particular picture is from the Greater Antilles mangroves. What is the thing is that when you talk about mangroves are always in combination with sea grasses, mangroves, sea grasses, and then coral reef. They are the very important component and they form a system. Mangrove has very important role because it locks the debris and it absorbs excess of nutrients like phosphorus and nitrogen from the water before it is actually released into ocean. And then sea grasses also no, they maintain a system. At the same time, since mangroves are plant-like structure, they also protect the coastal area from tsunami and high waves of ocean or sea. Sea grasses also absorb lots of nutrient and then they form a system which found ground for various species bacteria to grow. And then the coral reefs, I must say that everyone is knowing what is the importance of coral reef. They have entirely a different ecological system. So these all three form a complex system which found a breeding ground or the nursery ground for the various fishes, and it also harbors good numbers of microorganisms that play a very important role in decomposition and biological cycle, eco-biological cycling of various matter. they are, if you talk about what they are, what actually they do, then they are nursery grounds for various species, for breeding ground for various fishes. They are home to many species, as I've just shown you, the ridley turtles, proboscis monkey, fish, climbing fish, and then kingfisher, bengal tiger. So they are home to many species. They also provide food for various microorganisms, which actually take food and then they decompose it, make it available for other organisms. They also work as a substance for cleaning of the water, because when you talk about mangrove, they are on the zone of sea and river. So what they do, that they are managed to demarcate the two types of water, and they do not allow the mixing of water, so that a gradient is maintained from the river to the ocean. So that is why, since they are plant, they are tree, they are rooted tree, they are able to maintain a stable coastline. They are sheltered from storms and tsunamis, and various resources are available which humans are exploiting for their livelihood and for other purposes. Now they are important because they are able to go for nutrient cycling. They are able to maintain the hydrological regime. They are protecting the coastal area. They are protecting fishes and various plant species. They are also act as shock absorber. They reduce high tides and waves and help prevent soil erosion as well. So they also provide livelihood opportunity to coastal communities, and that is why they are very important. If we talk about the human activities, since deforestation has caused various climatic changes, and one of the important climate change is the global warming, and because of this the sea level is rising, and this has also caused reduction in fresh water. As why? Because there is huge amount of fresh water is being flown because of the melting of the ice. So this is causing a continuous annihilation of the mangroves, and it is estimated that the way the climate is changing in the coming years, 10 to 15% of mangroves will be lost, and this is very so, challenging to us, because at the time when we are looking for human civilization development, it is coming at a cost. And what is the cost? The cost is that we are changing this climate, which is ultimately changing the entire ecosystem on this earth, and ultimately once the system will change, this earth will change, it is going to change our life as well. So this is a very important issue to understand, that what is actually we are supposed to do. See, we do a lot of activity which is actually killing this environment, and so what we have to understand here, that we have to change our attitude. We have to be in harmony with the nature. We have to understand, like the animals, if there is if they are in need of something, then only they go for example, if a tiger is hungry, then only he will run. But unlike animals, we human, although we have more wisdom, we are more intellectually strong, but still, even if we are not hungry, we will still be fighting, we are still be running behind to assemble, to gather lots of resources with us. So that is the one important thing that we need to change, that we have to be in harmony with the nature. We have to learn from the nature, that how to be in sync with the nature. The second thing, that we have to act as a component, not as a master of the system. Actually, we always use the word exploitation. In fact, it should not be exploitation. It should be that we are using for a common purpose. So the thing is that we always think that whatever it is available in the nature, it is for us to consume. Rather, it is not for us for consumption. It is for us. It is available for us to use. So we have to understand that we are actually a component of the system. We are not here as a master. And ultimately, if I always take this example, if this earth can really throw big animals like a dinosaur or the mammoth, then human being is very small creature in comparison to what nature has the capacity. So if at all we are not going to curb our ways, we are not going to mend our ways, definitely with nature will throw us one day. And that is I have written that we have to curb our policy of use and throw. Whatever we take things from nature and then use and then throw it like anything. it actually creates problem for us, because while taking from nature, we are actually depleting, we are every day we are decreasing the natural resources, and while throwing in the environment again it is causing pollution, that is, it is creating the resources to degrade and deplete both. I mean, it is degrading the resources, at the same time it is depleting also. That is a dual problem we are creating with the policy of use and throw. So what we are supposed to do, that we have to adopt the policy of 3R, that is reduce, reuse and recycle, which will ultimately cause the little less pressure on the various resources, natural resources. At the same time, it will give us a time for replenishing it, so that for rejuvenation, replenishment, so that whatever loss we have done, the earth can naturally come up with that losses, and it can again become a more habitable place. And this corona, this lockdown imposed corona has actually taught us that in a very short span of time, once we stopped using our vehicle and stopped working with factories and all that, how the environment has cleaned in a very short span of time, and it shows some miraculous views that we have not seen for ages. Some of you might have noticed that you have observed clear sky for the first time in your entire life. So this is the time when you need to understand that we actually have to curb our ways to maintain various resources. Mangrove happens to be one very important resource, because it is maintaining the all marine ecosystem. And if you talk about the marine ecosystem, perhaps it is the largest ecosystem which is available on the earth. So we need to really protect this ecosystem, and mangrove, coral reef and seagrasses are acting as a complex shock absorbing system which actually protect our marine ecosystem. So with this, I need to conclude here. This is at high time that we curb our ways and think on those issues where these forests or these animals or these plants are living. We need to protect their houses as well. Thank you so much.
Thank you so much, ma'am. This actually gave us a basic outlook about mangroves and the importance of this day. So let's make the center stage ready for the chief guest of the day, Dr. Kundan Kumar, who is an associate professor at Department of Biological Sciences at BITS Pilani, Goa campus. He was also a postdoc fellow at McGill University for 6 months in 2011. He was also a postdoctoral research associate at University of Massachusetts Amherst for another 6 months. Then he is a PhD in plant molecular biology from Jawaharlal Nehru University. So we can conclude his area of interest can be plant molecular biology, plant philosophy, plant signaling, plant biotechnology, and plant natural products. He is also working on mangrove species in Goa for last 6 years. So let's not mention too much and let his knowledge talking. Sir, you can please start with the workshop now.
So thank you so much for introducing, as well as I would like to thank Dr. Nivedita for asking me. She has she has basically given me very short notice to prepare the slides, and already she has told you about some of the things about mangroves. So I will just and also I would like to thank Director Sir for a brief introduction about mangrove plants and mangrove ecosystem. So as Dr. Nivedita asked me to a little bit brief about mangroves. Already she has briefed little bit, and I will just little bit brief about it. And also I would like to just tell you about my work, which I was doing for last 7 to 8 years. So basically, so I will introduce mangrove plants. Mangrove plants is basically, you can see that they are basically, you can see that they are ecosystem. They are not only plant. There are other animals are there, the other fishes are there, and also various types of organisms live there. So, but mangroves is called because of the type of plant which is present in that particular ecosystem. So mangroves are basically called because of the plants, not because of other organisms. So here the title of my talk is mangrove plants, their molecular identification. So molecular identification on basis of some of the markers and their bioactive properties. So bioactive property in sense that whether they are having some natural properties to cure some of the whether they have some antimicrobial properties or they have some antioxidant properties or anti-cancerous property. So that we are going to see in my further slides. So, what are estuaries? Estuaries can be defined as a standing water body within a coastal region where there is greater net inflow of fresh water than an influx of seawater, right? So estuaries is a place where most of the mangroves vegetation lies, and this is the area where river water as well as sea water will basically they will mix up, and here the water level gets rise and fall with the tides. So here salinity fluctuates. Basically, salinity goes more than sometime 100 ppt, and sometime it goes less than 30 ppt. So salinity basically fluctuates in that particular area. And as you know that estuaries are most fertile ecosystem in the world. So most of you are, as he told me that you're from BBA or MCA, but you must have read all this information from EVS and from science, because some of you may have read biology up to 10th. So you may be aware about this. So I think that you are aware of estuaries, that they are most fertile ecosystem in the world, and there is a greater productivity is there. Even more than the adjacent ocean or freshwater upriver. So estuary is a place where the most of the mangroves grows. And here in Goa region, so there are like two estuaries are there, Mandovi estuaries as well as Juari estuaries are there. So most of my studies was focused on those estuaries, and we go there, collect the plants, and we work on that. So you see that these are the plants which are called as mangrove plants, right? They are growing near the estuaries. And these are the plants which are already talked by Dr. Nivedita, that they have different type of root system. Prop root system is there, as well as some of the those nematophore type of root system will be there, that we'll see, right? So these are the mangrove plants which are growing near the estuaries, and they are basically grown in shallow water. They are not growing in the main estuary, but they are growing in the shallow water. And you can see that this mangrove forest is also called as mangals. And they are not only limited to estuaries, they are also present in the salt marshes, and they sometimes they coexist, right? So mangroves are flowering land plants, and they are adapted to live in the intertidal region. So you can see that these are the mangroves which are just outside the particular mangrove forest area. So you can see the prop roots are there. You can see stilt roots are there here. So these are the stilt root. So basically these mangroves are called as red mangroves, because they are basically when water current will come, these are the first mangroves which will these are the first mangrove plants which will just prevent the tides, right? And they are mostly grown in higher salt concentration. So these are the red mangroves, and the one which is growing just after this one, that will be called as black mangroves, right? So they will have this type of nematophores. You may be knowing that some of the mangroves they have prop roots, some of the mangroves they have nematophores, right? So these are breathing roots. These are also prop roots are also breathing roots, because they have the lenticels through which the gaseous exchange can take place. And these are the breathing roots. These are the nematophores. These where the gaseous exchange takes place, because these cells. These nematophores they will have a lenticels through which the gaseous exchange can take place. So most of the nematophore type of roots will be found in black mangroves, and the prop type of roots will be found in red mangroves, right? So so basically we will see that mangrove forest has been rapidly destroyed by humans. So basically mangrove forests are getting destroyed because of natural processes, because of some tsunami, or if some high tide comes, then also it can cause damage to the mangrove forest. So these mangrove ecosystem is very fragile because of those conditions which are there, high salt concentrations. And so why these are very important? Because these other than mangrove plants, no plant can grow in this particular region because of high salt concentration. Because of high salt concentration, none of the other plants can grow here, right? Only mangrove plants can grow here, or some halophytes can grow here. But halophytes also, they can tolerate high salt concentration, but they cannot tolerate high tides. So that is why mangroves are very important, because they can tolerate high tides, they can tolerate high salt concentration. So all these things makes them a system where they can prevent, or they can stay in that particular region. Otherwise, apart from mangroves, none of the plants can grow there, right? That is because of high salt concentration, as well as the tides, as well as some harsh condition which is there in that particular region. So what are the unique features of mangroves? Some of the unique features of the mangroves, as you have seen, that the prop roots or supporting roots will be there. So the prop roots or supporting roots will be there in the red mangroves, which is just outside the forest area. So most of the mangroves, if you see this type of mangroves, these are the red mangroves. Basically, red mangroves they belongs to Rhizophoracea family. Rhizophora imicornata, Rhizophora apiculata, they belong to this. So you can see this type of roots are there, prop roots are there. So these are basically these are the red mangroves, and they are basically the main mangroves because they are tolerating higher salt concentration than the other plants which are growing just after that. So, but you can see that the clear-cut difference between red mangroves and black mangroves is that there is a supporting roots are there like this in case of red mangroves, and in case of black mangroves, which is growing after this one, you will find that there is a nematophores are there, nematophores which are breathing roots that will be there in the black mangroves. Right. So these are the two mangroves plants you can differentiate on the basis of their roots. Right. And other thing is that they have a salt excreting leaves are there. So you can see the salts here. You can see the salts here, which has been excreted out from the leaves. Because of that also, they have a salt exclusion mechanism. They have salt excreting glands through which the salt will be excreted out. And that is why they are able to grow in higher salt concentration. So this is the main adaptation for the plants. Because of that, they are growing in this particular region where the salt concentration is almost. So salt concentration doesn't basically it is stagnant. Sometimes because of high tides, when high tides comes, then the salt concentration goes more than 100 ppt. And in case of low tides, the salt concentration is less than 30 ppt. Ppt is parts per thousand. Right. So if it is less than 5%, and then also they will be able to grow. If higher concentration of salt is there, then they will be also growing. So most of the time, higher salt concentration is done. It is basically taken care by most of the Rhizophoracea family. Rhizophora, basically Rhizophoracea family. And then another unique feature of the mangrove plants is vivipary. Vivipary means the seeds will start germinating. You can see the seeds are getting started germinating when they are on the plants only. Because when seeds will not germinate and they will fall into the water, they may not germinate there because of the particular conditions may not be there. So the vivipary is the process through which the seeds will start germinating when the seeds are on plant itself. They are attached to the plants. So after germination, when they will fall, they will find some surfaces on that they can grow themselves. So these are the four major characteristics or features which is present in mangroves, that is supporting roots, nematophores, vivipary, and salt excreting leaves or glands. So there are, as I told, that red mangroves, black mangroves, white mangroves, there are various types of mangroves are there. So most of the cases you will feel that the red mangroves are the more vulnerable because they are on. So in a mangrove forest, you'll find that red mangroves are towards the most water side, and they will have prop roots. So most of the time they will be inundated in the water, and they will have more salt concentration. They will be they are having more. They will feel more tides, and they will feel more. They will have more salt concentration. They will have and after that black mangroves are there, where you will find the low. So basically, they will be after red mangroves, and here the difference between red mangroves and black mangroves is that you will find here prop roots, and here you will find the nematophores. Right, and here the salt concentration is a little bit lower because they will absorb most of the salt. Right, and white mangroves are the one which is basically Lagunicaria racemosa are the plants which are coming under white mangroves, and they can tolerate very less concentration salts. So after black mangroves, these are the mangroves will be there, and these are the associated mangroves, and where the salt, they cannot be able to tolerate higher concentration salts, but they are also grown in the mangrove vegetation, buttonwoods, but they are not basically true mangroves. So mangroves can be decided. So basically, these are the true mangroves: red, black, and white mangroves. Buttonwoods are not true mangroves; they are associated mangroves, right? So, what are the importance of mangroves? As Madam has told you, and Sir also told so many things about importance of mangroves. So, I'm not going in details. So, they have complex root system, and and basically you can see that they are the shelter. They basically they absorb most of the pressure from those tides, high tides, and all. So they have a protective role, and it has been widely recognized, especially after devastating tsunami of 2004. That they wherever more mangroves were there, they were that much devastation was not there compared to where the mangroves were not there, right? And also they are basically they arrest coastal erosion and seawater pollution. They are fertile breeding ground for many fish species and other marine fauna. They are the important source of livelihood for various coastal communities, and most of the people are dependent upon their medicinal properties, honey, tannins, waxes, and fishing. So these are the livelihood for various coastal communities, and in Goa also various community depends upon this particular mangrove ecosystem. And they are important carbon sink because you know that a lot of plants are there, so they can so whatever the carbon dioxide will be there, that can be absorbed because of they have higher photosynthetic rate, and that is why they are important source of carbon sink. So you can see that in mangrove ecosystem, not only mangrove plants are there, there are crabs there, skippers, sponges, polychaetes, mud shrimp, clams, and most of the time you'll find that birds are nearby mangrove plants or mangrove ecosystem, because and so many places you will see that there is in Goa also they have created a Salim Ali bird sanctuary where people used to go in the mangrove forest and they can watch birds, because these are the natural home for the most of the birds. So that is why birds, and it is good point for tourism, as well as it is having a bird watching also what person can do, right? So this is a mangroves is a plant, but it is an ecosystem also because so many things are associated with mangrove plants. So if you see the world distribution of mangrove forest, basically it is present between 30 degree of north to 30 degree of south latitude. They are not present after that. Okay, so they are present in this area only. They are present in this area only. They are not present in after the 30 degree north and 30 degree south, right? So they are present between 30 degree north to 30 degree south, right? So mangrove forest are, so that is why most of the time the countries which are mostly having mangrove vegetation belongs to Indonesia, India, and most of the time it is the South American countries. They have most of the mangrove vegetation compared to any other countries. So if you see the world mangrove distribution, most of the time the East Asia constitute about 33.5% of total world mangrove distribution. After that, it is South American countries where 15.6% of mangrove vegetation lies there. And South Asia, it is about 6.8%, out of which India constitute about 45% of total mangrove from South Asia. Right. So these are the world mangrove distribution, which suggests that these are the areas where most of the focus should be there. So to conserve the mangrove vegetation, because you know, because of natural anthropogenic processes as well as natural processes, the mangrove vegetation is shrinking. Okay. So if you see the global change in mangrove coverage, so if you compare 1918, where the mangrove coverage was about 45 million hectare, right, for 50,000 hectare in 1918. So from there in 1970 we have 288,000 hectare. In 1980 it came to 175,000 hectare, and in 1988 basically it is 138,000 hectare, right? 140,000 hectare, and now currently in 2020 it was demonstrated that it was we are having 150,000 hectare. So if you see, the mangrove vegetation has gone down from 450,000 hectare area. The mangrove vegetation area has gone down from 450 million hectare, 450,000 hectare to 140,000 hectare. And now in 2020, it was demonstrated, it was shown that 150,000 hectare is there. on which the mangrove vegetation is there. So quite 3 to 4 times reduction of mangroves has been taken place if you compare from in 100 years. So if this will continue, then it has been shown that in 2030 we will have less than 100,000 hectares of land with mangrove vegetation. That is the main issue, why we should work on towards the reclamation or restoration of mangrove plants. Because of you see that because of natural processes as well as various anthropogenic processes, the mangrove vegetation is getting lost. And we have to reclaim those mangrove vegetation. And India is the only country where you can see that last time 2017 the mangrove vegetation was about 47 million hectare, about 4.47 million hectare, and after that we reached to there was some increase of mangrove vegetation happened in last 3 years. It has been shown about some 0.1% or 0.2% of increase of land area on which mangrove vegetation was there, that was increased. So, but apart from that, if you see the world mangrove coverage, it has decreased a lot. So, in case of Indian mangroves, so World Atlas of Mangroves shows that there are 73 species there in 123 countries. So out of 73 species of mangrove plants, mangrove species, 46 is there. 46 species are present in India. Okay, and that too not in all the states. Delhi doesn't have any mangrove species, right? So it belongs, it basically because of they are present only in estuaries. So it will be present only in those areas where the coastal areas they will be present only in the coastal areas. So they are present in 9 states and 4 union territories of India. Right, so you can see the highest number of land area where mangrove plants are grown, that is in Sundarban, right? And after that, Gujarat is there, where the second largest land area is there for where the mangrove plants are growing, right? So if you see the mangrove distribution in India, there's like, as I told that there are about 12 states are there, 12 states and union territories are there in which mangrove vegetation is growing. And most of the area in which the mangrove vegetation is there, that belongs to West Bengal. And this is like in largest mangrove vegetation is present in Sundarban. World's largest mangrove vegetation is present in Sundarban. So mangrove covering state, if you see, as I told that most of the mangroves is present in West Bengal, but you can see that there are some other places also like Gujarat, 23.6% of mangrove is there. And then Andaman Nicobar Islands, about 12.3.9% of mangroves is present. And Goa is a very small state where 0.52% of total mangroves area is there in Goa, right? So, but you see that the mangrove speciation, there are about 43 mangrove species is there, but Goa is very diverse because of even though the area is small, 0.52% of area is there for mangrove vegetation, but number of mangrove species is about 14 we found. Total number of mangrove species was present in Goa is about 14. So next slide, please, ma'am. Yeah, so these are the threats which mangrove faces. The threats includes climate change. So because of rising sea level, this will increases soil salinity, and that will also reduce the growth of mangroves. And also agriculture, because most of the area will be utilized for agriculture, so the mangroves area can be cleared up. Then coastal development, because of infrastructure development, business and hotels and homes also, you can see that coastal area is getting shrinked. Over harvesting of mangroves for firewood, wood, charcoal and animal fodder also, that can reduce the mangrove, that is also becoming a threat to the mangrove ecosystem. Overfishing can also, because there is a ecosystem is there, and that will be disturbed if overfishing will be done. And aquaculture, mangroves destroyed to create space for fish pond, that can also reduce the mangrove vegetation. So these are the threats which is faced by mangroves, and we have to see that if we can work something towards restoration of those mangroves, and we should not allow the polluted water to enter into estuaries, or we should not allow the other system to get into the mangrove ecosystem, and we should not do the infrastructure work, because if you see in Goa conditions, the roads are getting widened up. So some of you, if you have visited Goa in last 2-3 years, the roads are getting widened up. Because of that, a lot of mangroves are getting they were removed from the particular places. and they were basically, even though they were removed, you have to restore it at certain places. So restoration is also required. So that is also one of the things which is required, and that we can participate, not only the biologists, the other people who are working towards the betterment of the ecosystem or betterment of the entire world, or particular India. One can think about restoration of mangrove ecosystem, and there you can grow more of the seedlings, and then you can restore those things. So what we are doing basically, what in our lab, what we are doing. So basically our lab is currently working on mangroves on two particular things. So first is DNA barcoding of mangrove species of Goa. So in Goa, there are, like I told, 14 species of mangoes is there in Goa. So we try to barcode those. Barcode means you can identify. So basically, the mangrove can be, they are very similar. Mangrove plants are very similar. So you cannot identify it on basis of morphological features, because the leaves are almost similar. So most of the species, they will have buttress root, prop roots, and those pneumatophores will be there. So it is difficult to differentiate one mangrove species with another mangrove species. So what we can do, we can go for sequencing of the particular mangrove species, and you can sequence those and you can find it out, or you can give some barcodes that this is particular because of the presence of the sequences. This is particular species of mangrove. Like that, we have done DNA barcoding. First time we have done DNA barcoding of mangrove, entire mangrove species of Goa, which is about 14 species are available. And second thing is that screening of identified mangrove species for its antimicrobial properties. So we have checked there, and so you know that these are the from long time the people of various communities which are dependent upon mangroves. They are basically they are utilizing this as a resource for medicinal purposes and all, and isolation of. So we also thought that it may have some medicinal properties. So why don't we see some of the phytochemical compounds and try to purify those compounds and to check that whether they have some antimicrobial properties or not. So we have tried to see this. We have to screen. We have screened entire mango species, and we found some of them are promising. Next slide, ma'am. So I'm not going into deep because now you don't have time also. So there are like barcode markers are there. Some of the genes are there which we called as DNA barcode markers. So in plants there are like different barcode markers called the RBCL and MATK. These are basically gene sequences. And on the basis of gene sequences, they are basically they are unconserved gene sequences. That's why they, on the basis of these gene sequences, you can differentiate one mango species with another mango species, right? So in animal system, there is only one barcode gene is there, COI, okay, cytochrome oxidase. But in case of plant, there are it's not confirmed that it is only two. Where basically people are working with so many types of DNA barcode markers, right? So yeah, so these are the mangrove species. Can you go back, ma'am? If you see the mangrove samples which we collected from Goa, that belongs to Avicenna marina, Avicenna alba, Avicenna officinalis. So all these basically these belong to Acanthaceae family, and these Brugueria cylindrica, Brugueria zimnorhiza, these belong to Rhizophoracea family, and this Aegicetus cornea. So there are about 14 different species are there, and which belongs to 6 different family, and most of them are basically tree species. And we collected the sample, we prepared herbarium. Next slide, ma'am. The herbarium is in this form. You can see the different forms of plants which is present, and you can see the most of the plants are similar. So basically you cannot differentiate this plant with this plant. So on the basis of so once you can see the flowers, taxonomists can identify the on the basis of flowers, on the basis of seeds, on the basis of type of leaves. So but on layman, but without flowering, one even for taxonomist also this is not difficult. This is very difficult to differentiate between Brugueria cylindrica and Brugueria zimnorhiza. Similarly, Rhizophora micronata and Rhizophora apiculata, they are mostly same. You cannot see the difference between them. So we try to see that if we can barcode them and try to, yeah. So these are the basically the we have sequenced the genes. And these are the accession number of different genes that belongs to RBCL, MATK, ITS region. And then go to the next slide, ma'am. Barcoding of selected species 20th. Yeah. So here you can see the barcode we have done on the basis of these 4 markers we have done. And we found that ITS plus RBCL and MATK is the best suited marker which can barcode entire mango species of Goa. So you can differentiate. You can basically on the basis of with the bioinformatics tools, we have differentiated all the mango species on basis of species as well as genera. The genera should be differentiated between different genera, right? And species should also be differentiated on the basis of with different bioinformatics tools, we have tried to differentiate the different type of mango species on the basis of these MATK plus ITS2 genes. And then we have tried to see try to say that this region, this MATK region and ITS region can be able to differentiate all the mango species of Goa, and that too 14 mango species of Goa, right? So this entire results we have published in 2018 PLOS One journal. Next slide, ma'am. So after that, after identification of mangrove plants, and now we can able to relocate that where the plants are, we are trying to see that what are the bioactive properties of mango species. So we checked the literature and we found that they are having, mango species are having antibacterial properties, anti-cancer properties, antifungal, and these are the compound. These are the plant species like Agiceras corniculatum. These are the bioactive compounds are there. And these are anti-inflammatory compounds are there. You can see there. So some of the work has already been done, but none of the work has been done on in the Goan mango species. And we thought that we should work with the Goan mango species and try to find out what are the antimicrobial compounds which is present in Goan mango species. Next slide, ma'am. So we have gone for antimicrobial assay. We have gone for oxalate extraction with different solvents, and then we have done MIC assay, phytochemical analysis, and then checked. And further on, we try to identify a single compound which having some potential properties. Next slide. And these are the antibacterial properties. You can see the ethyl acetate extract is showing the inhibition. So the Avicenna marina, this is AIM is Avicenna marina, Acanthus. So basically AC is Acanthus ilicifolia. So basically this is, so here in the wells we have added the crude extract and we found that the bacteria is not growing. That means it is having antibacterial properties. Similarly, we have seen here also Rhizophora micronata. So you can see Sonarchia alba. So these are the organisms we have placed, like, so it is for Streptococcus aureus, Streptococcus epidermis. It is for Xeromonas aeruginosa. So they shows that this particular mangrove extract, they have some antibacterial properties. Next, ma'am. So these are the antibacterial properties of methylonic extract. So we have extracted in different solvents and we have tried to see different types of solvent is having different properties. Next slide. And these are the zone of inhibition. So the highlighted color shows that highest zone of inhibition against different types of organisms. So Streptococcus aureus shows highest zone of inhibition from Avicenna marina, right? And Bulgaria gymnorrhiza also show highest zone of inhibition. So this plant can be used against this Avicenna marina and Bulgaria gymnorrhiza plant can be used against Streptococcus aureus or Streptococcus epidermis, right? So we also checked for anti-candida activity and we also find that they also inhibit the crude extract. We have not purified it till now. So we have checked anti-candida activity and we found that some of the mango plants shows anti-candida activity, right? And these are the zone of inhibition you can see. So these are the, and next slide, ma'am. Yeah, these are the various phytochemicals which is present in mango plant extract. So we have, these are the different solvents like hexane, ethyl acetate, ethanol, methanol, and these are the compounds like saponins, phenolics, flavonoids, alkaloids, tannins, and terpenoids. So this is a qualitative essay where try to find out which are the compounds are there. So we found most of the plants is having tannins, they are having phenolics. So most of the compound was having phenolics and tannins. Next slide, ma'am. So here you can see that the Sonarchia cacellaris, they have a higher amount of total phenolic content. So total phenolic content was higher in terms of 196 microgram per GAE per gram, right? TFC is basically a total flavonoid content. So total flavonoid content was higher in Bulgaria gymnorrhiza as well as Sonarchia cacellaris, right? So next slide, ma'am. So these are the polyphenolic compound. We try to see that whether these are, so these, some of them are having show the antibacterial properties. So we have tried to find it out. We have quantified them with the help of LCMS facilities. And these are the like chlorogenic acid. So these have been detected in negative mode. So these vanillin and coumarin has been detected in positive mode. So I'm not going into detail because you may be not familiar with these topics. So I'm just going through it, right? So these are the chromatograms. So if you do LCMS, then you will find different. So you can see the one single peak is there, which belongs to chlorogenic acid. So we have a standard, and we found, and on the basis of that, we can quantify them. So what is the concentration of ferulic acid in that particular plant? So next slide, please. And this is positive mode, vanillin and coumarin. And subsequently, we can do vanillin. We can quantify vanillin content and coumarin content in plants. And next slide, ma'am. Yeah. So here you can see that Agiceras corniculatum, they have higher chlorogenic acid. These are phenolic compounds. Then caffeic acid is highest in Avicenna officinalis. So we have checked entire all 14 mangrove species. What is the phenolic compound? Different phenolic compound, which are like 10 phenolic compound we tested, and we found the differential content of those phenolic compound. And this phenolic compound can be used as a this phenol compound can be used as a antimicrobial properties. So we have also checked that whether this particular phenolic compound can be used as antimicrobial properties. Next slide, ma'am. So these are the list of compound detected in selected plant extract by GCMS. We have done GCMS analysis, and further on we have checked with the LCMS analysis, and we have found different types of phenolic compound, different type of secondary metabolites in plants, and these are the areas which suggest that they are having higher concentration. Next slide, please. So these are the LCMS screening. Next slide. Yeah, and then we try to find out, identify some compound which are basically, we try to purify this on the basis of TLC, on the basis of different purification components. We have tried to purify it, and after purification we checked. So our target was one bacteria. We found that it is showing very nice result. Next slide, ma'am. And we have purified it on basis of that, and we found that this particular bacteria is very much so with this is earlier whatever I showed you, the MIC assay or the inhibition assay, that was for crude extract, but now this is for purified compound. So purified compound shows very nice antimicrobial properties against Pseudomonas, and we found the zone of inhibition was about 30 mm of which can be considered as very higher zone of inhibition and very higher antimicrobial properties shown by this particular purified compound. Next slide, ma'am. Then we purified it by help of preparative HPLC. So I'm not going into details. Then we have characterized this compound. We have characterized this compound by IR spectrophotometry, and these are TLC based examination of isolated compound. We have used N-acetaldehyde, ninhydrin, and also KMNO4. So these compounds, these particular reagents was used for checking that what are the component, so that we can check that what is the compound may be. And we have done some physicochemical characterization, physiological characterization, which is shown in the next slides. Yeah, ma'am. Next slide. So this shows that effect of this is the effect of isolated compound on bacterial growth. So when this is all with the purified compound. So with the help of purified compound, we showed that the bacterial growth was reduced if we are adding more amount of the identified or purified compound. And this is the optical density, and we have checked that the relative gene expression, which is of Pseudomonas aeruginosa, which is related to this pathogenic activity. And we have used control samples, then treated sample with the particular isolated compound, and with salicylic acid, which is the positive control. And we found that the genes which are responsible for virulence genes, they are getting deactivated, or they are getting reduced when we are treating it with some of the higher concentration of purified compounds, and which is related to the salicylic acid also. Next slide, ma'am. So these are the some activity assays we have done, the virulence factor analysis on anti-biofilm activity, rhamnolipid, pyrrolidine, which suggests that this compound is having the properties of antibacterial against Pseudomonas aeruginosa. And the conclusions of the presentation which is made is the next slide, ma'am. So as I've shown you that our result shows that MATK and ITS2 can be helpful to demarcate mangrove species at mangroves of Goa at the species level, right? And mangroves are rich source of antioxidant, phenolics, and antimicrobial properties. And the ethyl acetate extract of mangroves are having very higher antimicrobial properties. And this particular Avicenna marina, Sonaricia cassiloris, can be the potent, they are the most targeted mangrove plants which can be used against the microorganisms. And the data from LCMS shows various metabolites which suggest that they have various bioactive compound where the more purification steps are required to purify the compounds and see their activity. And the isolated antimicrobial compound having bioactivity against bacterial isolate activity guided was used to isolate the bacterial compounds and purified using column and preparative HPLC. And this isolated compound shows that the active fraction is, and so we are trying to see what is the structure of that compound by NMR, by nuclear magnetic resonance spectrophotometry, and then we will try to see that if we synthesize that compound, then how nicely we can kill those organisms, and that will be our target to see. And these are the lab members. Mostly this entire work is done by Nilesh Dahibate, and the collaborators are Professor Utpal Rai, Rahul Janamade, Dr. Devendra Kumar, and Dr. Sudhakar Srivastav. And these are the funding support I received in last 9 to 10 years. Thank you. I will be happy to answer the questions if you have any.
Can I ask a question here, Dr. Kundan?
Yes, ma'am. Yes, please.
Yes, sir. What the presentation? It was an excellent presentation, and it has opened a new, discussion topic for us. Whatever we discussed up till now, whatever we were knowing that it is very important ecologically, but now you have shown us how they are important and how again they can be used for treating various bacterial diseases or fungal diseases. So that is absolutely something very surprising for us, and just wanted to understand that have we actually commercialized any of the product, not from Goa but from any other mangrove forest in India?
Basically, some of the groups has worked on it and they identify it, but it's not commercialized till now. Any product has not been commercialized, but people has purified and showed that some of the compound they have like as I told, that some of the compound listed in the first slide where it was shown that some of about 10 to 12 compounds are there, which shows various types of activity, and they have identified and purified the compounds as well. But commercialized purpose, you can see that there may be some other product in the market which may not allow this type of compound. So if they should be very much competitive against those particular things which is already available in the market. So that is what the thing, like it may be that is why it may be not in the commercialized state. But yes, some of the compounds has been identified and they have been purified and they have been shown that they have some antimicrobial or anti-cancerous compound. So till now we are in the process of identifying the compounds or elucidating the structure of the compound. So till now we have not. So if we find the structure and all, then only we can do the patent and then we can go ahead with commercialization. But from entire Indian perspective, I can say that till now the compound has not been commercialized till now.
It also demands that lots of activity is required and lots of stakeholders should come forward to promote such type of research, and really they should fund it so that at least they can be developed. And as you said that you are about to patent it once it is your structure, you are able to find the structure. So this is one why I'm asking, because post-COVID lot of options have been opened, particularly in health and pharma industry. So anything which is coming to support your health system or health, the general health of the people, then it should be really worked upon, because now it is a time COVID has taught us to work on health system. So that is why I asked this question. Now the second question is, sir, that See, most of the time, the other use, or rather the word exploitation, is that we can also use these plants for, these forest species for, revegetation, or the, once just reclimatization of the destroyed system. So I just wanted to understand these property, that antibacterial or antifungal properties of these plants, are they going to help the plant to restore a destroyed ecosystem?
So that is also a question mark, that if you isolate a compound and then you have to, if you isolate a compound, then you have to synthesize it and then you can use it, because forest department will not allow you to harvest that much, because you know that for if you want to isolate a compound and if you want to commercialize it, you have to have you have to harvest entire plant, or entire vegetation of that particular will be harvested. So the thing is, like, if you want to use it as a for pharmaceutical compounds, so that is what I heard from CDRI. There's a company, there are people, group of people are working in CDRI, where CDRI is Central Drug Research Institute. So where they have patented it, and but it was not commercial. It was they were not able to commercialize it because of this reason, because forest department will not allow to harvest most of the leaves from there, because this entire thing is based on so much of extraction, so much of so you have to synthesize it. So once you find if it is a small compound, you can synthesize it in lab and then you can see it. You can use it for commercialized purpose, but not from. So this will basically, if you harvest the plant for medicinal purposes, there is no the plant will not be benefited. Basically, it is for human consumption only, or for animal uses only. So plant will not be benefited because you are harvesting so much of leaves. So that is not required. So that is also why the pharma companies, they cannot take up this entire processes and they can move forward. So that is one thing is there, that you have to see that if you can synthesize this thing, whatever the compound you isolated, and synthesize it in laboratory condition and then use it for commercialization. But if it is a long molecule, if it is a long molecule where the synthesis will not be synthesis will have a lot of issues, then this will this cannot be done. This cannot be the pharma company will not take this as a lead and they cannot work on it.
Yes, sir. Thank you so much. Very rightly you said that yes, once you know the structure, you can synthesize it, and depending upon the process of the synthesis, the pharma companies will take or will not take. This is something absolutely right, sir. No, the other question: what happens with the destroyed mangrove forest? Are these compounds help them for any sort of reclimatization of the destroyed mangrove forest?
So there may be.
Just to give a understanding what I am trying to ask. See, we have heard about that they are mycorrhizas. They help the plant for revegetation. So similarly, these compounds, are they going to help for reclimatization also for any particular destroyed mangrove forest system?
So see, this we are trying to isolate from leaves. So there may be some endophytes will be there, or some
Okay.
organisms will be there that can help them to reclimatization and all. But as such, we have no idea that whatever the compounds we are looking from the leaves, that can be
Okay.
utilized for reclamation for the mangrove station.
Okay. So whatever you have studied, you have extracted from leaves.
Yeah, leaves.
Yes. Yes. Okay. Thank you so much, sir. Thank you so much.
Okay, thank you for attention, and I hope that it was. It's now lunchtime for everyone, I guess. So if any question is there, let me know. Otherwise,
Yes, sure. Vanshika, please spit up.
Thank you so much, sir, for such unparalleled information. Like, we celebrate these days every year, but still we remain unaware. So we are looking forward for more such events and information. Thank you so much, sir, and thank you so much everyone for participating.
Yes, thank you
Dr.
so much.
Kundan, thank you so much.
thank you, ma'am. Thank you for invitation, and thank you for inviting me so that I had presented my work.
And I know that you are not, you have taken a second dose of vaccine today. And you gave me your permission that yes, you are available just a call away. and I informed you of very late. Sorry for that inconvenience, but still, you were very prompt and very gracious in making yourself present for today's program. And last but not least, I would also like to give thanks to Dr. Ajay Kumar, the director of the college. It is only his guidance that we are able to do such program here. And now he has also, Dr. Kundan, as I told you, that he has also given your candidature to the Delhi government for further such programs. You may be impaneled, and other colleges may be calling you for certain other programs. So thank you so much, Dr. Kundan.
Thank you, Dr. Nivedita. Thank you so much for inviting me.
Yes, please. Thank you, sir. Dr. Bajpayee sir, thank you so much. आप भी हैं? Dr. Bajpayee sir.
Yes, madam. I am there from the very beginning. I am listening each and everything.
Sorry sir, मैंने देखा नहीं. How was sir? आपसे मैंने बुलवाया नहीं sir.
नहीं, मैं तो इसलिए आ गया कि यहाँ पे बहुत अच्छी चीज आज ये फ्लोरा फोना पे डिस्कस हो रहा था और मुझे भी याद आ गया अपना 25-30 साल पहले पढ़ाई छुड़े हुए तो बहुत टाइम हो गया ना. 11th में पढ़ी थी, 11th के बाद नहीं पढ़ी थी बायोलॉजी. जी. आज फ्लोरा फोना वर्ड सुन करके बिल्कुल पुरानी यादें ताजा हो गई और बहुत अच्छा सेशन था. एक्सीलेंट, बहुत बढ़िया. और थैंक यू डायरेक्टर सर जिनके इनिशिएटिव से और जिनके डायरेक्शन इनिशिएटिव तो हम लोग से कराते हैं सर, लेकिन डायरेक्शन सर का रहता है. बहुत अच्छा रहा जी. थैंक यू एवरीबॉडी.